I've said it before and I will say it again, free-base testosterone really doesn't matter if your androgen receptor density and sensitivity is suboptimal. Only the truly competent recognize the health of your ARs is the model in which you unlock your masculine gene expression, the true potential of our free-base testosterone. The truth is that many are still fecklessly unaware of this major component when they seek to realize their true masculinity. Since I've known this to be true, it's been my aim to realize what are the most powerful AR enhancing practices, which in my estimation is roughly between 6 and 8. But not only addition, but subtraction as well. In essence, what you should stop yourself doing in order to protect your ability to project your masculine genes. And fundamentally, how do I measure or quantify true AR optimization? What will I experience in my consciousness, in my body, when my masculinity is realized? When testosterone or DHT starts to flirt with an androgen receptor, it's like a flipping of the switch on a genetic power grid, lighting up all sorts of biological processes. On the grand stage, this means men start to channel their masculine genes, their beards, their sturdier jawlines, their bulkier muscles and trimmer waistlines. They also find themselves more temperamentally disposed for sports, competition, better at navigating social situations, and increase in their sexual power, libido, vitality. On the microscopic level, this molecular process inhibits stress-related neuron death, boosts your muscle protein synthesis, revs up the metabolic rate, kickstarts red blood cell production, and puts brakes on estrogen and aromatase. It is the process by which you realize your inner savagery, the hunter archetype that allowed you to cut through so many nameless men in order to be here. Androgen receptors in and of themselves are like the VIP lounges of your cells, only opened by exclusive members such as testosterone and dihydrotestosterone. In the grand concert of biology, these nuclear receptors are the maestros, conducting proteins inside cells, orchestrating vital functions like growth, regulation, energy expenditure, cell life and death, and division of those cells equally. Think of them as sophisticated lock and key systems where only the right keys, being the androgens, can unlock the powerful transformations within your body. Now, here's something I've already been doing that has, to my pleasant surprise, already had a notable impact on my AR sensitivity. So, androgen receptors seem to treat testosterone and DHT like long lost friends or twins once you break a fast. 2 illuminating studies highlighted this phenomenon. The first one was a Swedish study that found that fasting between 12 and 56 hours can amplify testosterone responsiveness by up to 180%. There was another intriguing study that, to be transparent with you, I'm finding difficult to source, which described participants undergoing a rigorous 10-day fast, and then they resumed normal eating habits. And their testosterone absolutely skyrocketed and continued to climb beyond the 5th day post-fasting period. So what might be the plausible explanation for that? Well, I believe their bodies become a little bit more sensitive and highly tuned to androgens during this grueling kind of calorie deprivation, making eventual reunion with food a cause for celebration, let's say. Now, I do need to mention on a personal note before you start fasting that long fasts, which I would consider to be 3 days plus, is something that I only do every quarter, so every 3 months. More realistically, my eating patterns kind of follow intermittent fasting, so 18 to 6, fasting for 18 hours, eating for a 6-hour window, OMAD, 1 meal a day, or alternate day eating. Because I believe there is some pushback that extensive fasting periods more frequently are going to be more catabolic than they are going to be anabolic. So I would do my own personal experimentation with this, and to be honest with most of these. A recent addition I've actually made that has gotten a lot of good research around it is L-carnitine and L-tartrate, which is typically found in red meat and fish. However, supplementation can further increase androgen receptor content. So listen closely to this very cool study. The study was aimed to determine the effects of a 3-week L-carnitine supplementation, we'll call this LCLT, and post-resistance exercise, or RE, feeding on hormonal and androgen receptor responses. Now, the methods involved 10 trained men, and they were given 2 grams of L-carnitine per day, or a placebo for approximately 21 weeks. And they provided muscle biopsies, so they went in, took some muscle for AR analysis, and performed 2 resistance exercise protocols, 1 followed by water intake and 1 by a meal, 56% carbohydrates, 16% protein, and 28% fat. Now, these trials were all randomized and included a serial blood draws and a post-resistance exercise biopsy. So, very comprehensive, and after a week-long washout, the subjects switched protocols. Now, you might be asking yourselves, like I am, what might be the results? So the LCLT supplementation increased pre-exercise androgen receptor content compared to the placebo. With resistance exercise alone, it only increased androgen receptor content in the placebo trial. Post-resistance exercise feeding significantly increased AR content comparatively to the baseline, and water intake in both LCLT and placebo groups. So feeding suppressed serum testosterone levels compared to the water intake and pre-resistance exercise values. Luteinizing hormone showed subtle changes in the responses to feeding and the LCLT. So in a more colloquial parlance, what does that mean? Feeding after resistance exercise increased AR content, potentially enhancing testosterone uptake and luteinizing hormone secretion. LCLT supplementation upregulated AR content, which may aid in recovery from resistance exercise, which means that 200 grams of L-carnitine should be something that you are including in your supplementation stack if you feel you're not getting that kind of amount in red meat and fish. Now, just a quick update on some fundamentals. So we are going to talk about resistance training being shown to significantly increase androgen receptor content in muscle tissues. Studies have found that trained men actually have higher androgen receptor content in their muscles comparatively to untrained, which shouldn't come as a surprise to you. More specifically, in the training modalities, heavy weightlifting with short rest periods between sets appear to be the most effective, combining heavy lifts with burnout sets, so to failure, at the end of a workout may aid in enhanced androgen receptor upregulation. And when you're training, you want to be training muscle groups which have the largest amount of mass with good form, and also some plyometrics, some explosive stuff, box jumps, sprints. That seems to also have a net positive effect on AR sensitivity. Additionally, we're going to talk about increasing protein intake will help boost AR receptor activity, and it will decrease sex hormone binding globulin while increasing free-base testosterone and IGF-1 levels. And this is important because it can stimulate metabolism and insulin response, which may in turn stimulate androgen receptor activity. Oh, and vitamin D. Additionally, you want to make sure you're getting adequate vitamin D levels, always associated with high levels of optimal testosterone and influencing the AR functionality. There's actually been mass studies showing correlations between vitamin D insufficiencies and low testosterone, and the antithesis is high vitamin D and, you guessed it, high testosterone. This next part is cool. So it turns out our rodent friends have literally spilled the beans on a little secret. Sipping on caffeine can give your testosterone and DHT levels a serious boost among androgen receptor expression. The magic trick: caffeine actually jumps into action by activating the CAMP enzyme inside the cells, housing these regulators. This then tags in the protein kinase, and you can think of this as the metabolic maestro that upregulates these receptors, making them super eager to bind with testosterone and DHT. It's like caffeine and forskolin were separated at birth, and both of these crank up the intracellular CAMP levels to get those androgen receptors reunited, I guess. So this next part, in my personal opinion, is why most men are comparable to women in recent times. I mean, that might be mad. Let's say not fully realizing their masculine potential. So research has demonstrated that sexual exhaustion can lead to a reduction in androgen-related density and immunoreactivity in specific regions of the brain, associated with male behavior. So a study done by Romano discovered that 24 hours after copulation to satiety, there was a significant reduction in AR sensitivity and density in the mediopreoptic area and the ventromediohypothalamic nucleus. So the same study indicated that 48 hours post-satiety, 70% of male rats exhibited complete inhibition of sexual behavior, while 30% displayed only one singular, very lonely ejaculation. This behavioral change correlated with a decrease in AR, specifically in the medial part of the MPOA. Now, research by Fernandez found that sexual activity diminishes AR receptivity, immunoreceptivity, in particular brain regions involved in the regulation of male sexual behavior. So the reduction in androgen receptors appears to be only temporary. So the Romano study did highlight actually after 72 hours of sexual satiety, the AR receptors do return to baseline, but there were still some problems in the other areas of the brain, such as the lateral septum ventral and the bed of the nucleus of the stria terminalis and the medial amygdala. However, of course, we have to highlight that 72 hours after abstaining from sexual activity is the precursor to healing the AR receptors. So stop touching yourself and you become untouchable. Next, there is a tropical legume called the Mucuna Pruriens. I'm probably butchering that. That contains high levels of L-DOPA as a precursor to the neurotransmitter dopamine. It indirectly influences AR function and testosterone level through its increased dopamine production. We all know there's a relationship there between dopamine and testosterone, and L-DOPA has shown to enhance the activity of ARs by acting as a co-activator in the protein in vitro studies. This means it can increase the sensitivity of AR receptors to the hormones like testosterone and DHT, potentially boosting their effects. Research in rats has demonstrated that high levels of L-DOPA, approximately 1,000 milligrams per kilogram of body weight, can elevate circulating levels of testosterone after 7 to 14 days of administration, which is likely due to its effects on luteinizing hormone. What may be additionally curious to note is in human studies, daily doses of 800 milligrams of L-DOPA for 7 days have been known to increase penile girth, if that's something that you want, especially when serum levels of testosterone were above 17.5 picograms per milliliter, indicating an androgen-dependent mechanism. L-DOPA's impacts on sexual functions is also notable as a consequence of the latter. It can enhance libido, sexual activity, likely through its dopaminergic effects. The effects of L-DOPA on androgen receptors and related functions are dose-dependent, however, with significant results observed at doses of 250 to 500 milligrams or more per day. Now, many studies utilize the Mucuna Pruriens, or it's actually called the velvet bean, which is something that I should be using a little bit more often, as my pronunciation is subpar here, as a natural source of L-DOPA for investigating these effects. So if this is of any interest to you, then the velvet bean might be something that you choose to tickle. But these are not theories today, gentlemen. These are facts. Speak soon.